The relationship between vitamins, autophagy, and longevity is far more than incidental. It is deeply rooted in the intricate signaling networks that maintain cellular homeostasis. While vitamins have long been viewed as essential cofactors in basic metabolism, emerging research increasingly positions them as active regulators of gene expression and cell signaling pathways, particularly those governing autophagy and cellular repair. This suggests that vitamins do not simply support life, they modulate its pace and resilience. A systems biology approach reveals how specific vitamins intersect with autophagy-related genes and molecular pathways that, when finely tuned, can extend health span and potentially influence longevity itself. Autophagy is a tightly orchestrated cellular process that enables cells to break down and recycle damaged components. It is vital for clearing out misfolded proteins, dysfunctional mitochondria, and invading pathogens. Autophagy becomes especially important in aging when the burden of cellular debris grows and regenerative capacity diminishes. But autophagy doesn't operate in a vacuum. It responds to nutrient availability, energy status, and oxidative stress, and it is regulated by a constellation of signaling networks including mTOR, YMPK, FOXO, SIRT1, and PGC1A. Many of these pathways are vitamin-sensitive. Take vitamin B3, niacin, for instance. It is a precursor for NAD, a molecule that powers sirtuins, particularly SIRT1, a key regulator of autophagy and longevity. SIRT1 deacetylates several autophagy-related proteins, including ATG5, ATG7, and LC3, promoting the initiation of autophagy under stress conditions. The NAD/SIRT1 axis is central to metabolic adaptation during caloric restriction and fasting, both of which are known to extend lifespan in various organisms. Without adequate B3 intake, or, in the context of NAD, depletion, which accelerates with age, this regulatory circuit weakens, compromising the cell's ability to mount an autophagic response. Vitamin D is another crucial player. Beyond its classical role in calcium metabolism, vitamin D functions as a transcriptional regulator via the vitamin D receptor, VDR, which directly modulates hundreds of genes, including those involved in autophagy and inflammation. Vitamin D enhances the expression of autophagy-promoting genes such as BECLIN-1, and it suppresses NF-B-mediated inflammatory pathways that impair autophagic clearance. Deficiency in vitamin D has been associated with reduced autophagic capacity, increased oxidative stress, and higher susceptibility to neurodegenerative and metabolic diseases, all of which shorten health span. Vitamin A, often overlooked in discussions of cellular maintenance, also plays a surprising role in the autophagy longevity axis. Retinoic acid, the active form of vitamin A, regulates gene transcription through nuclear receptors like RAR and RXR. This regulation extends to autophagy-related genes involved in lysosomal function and protein degradation. Retinoic acid has been shown to influence the expression of TFEB, a master transcription factor that controls lysosomal biogenesis and autophagy flux. Proper activation of TFEB enhances cellular clearance capacity, a function that becomes increasingly vital with age as intracellular waste accumulates. Similarly, vitamins B6 and B12 participate in 1-carbon metabolism and methylation processes that impact gene expression patterns, including those of longevity-related pathways. Impaired methylation may alter the transcriptional activity of autophagy genes and promote genomic instability. Over time, this contributes to age-associated cellular dysfunctions, from mitochondrial decay to increased DNA damage, conditions that might otherwise be mitigated by robust autophagic activity. Vitamin C, known primarily as an antioxidant, supports autophagy indirectly through its role in maintaining redox balance. Oxidative stress is a double-edged sword. It can trigger autophagy when mild, but inhibit it when excessive. Vitamin C modulates the redox-sensitive regulation of AMPK and mTOR, ensuring that autophagy is neither overactivated, which may cause excessive degradation, nor suppressed, which allows damaged organelles and proteins to accumulate. In aging cells, maintaining this oxidative equilibrium is critical for preserving homeostasis and avoiding cellular senescence. Systems biology also recognizes that these vitamins interact in complex, networked ways. For example, magnesium acts as a cofactor for enzymes involved in vitamin D activation and in ADP-dependent autophagic machinery. Zinc supports the structural integrity of DNA-binding proteins, transcription factors, and lysosomal enzymes. Deficiency in one micronutrient can disrupt an entire cascade, diminishing autophagic efficiency and increasing vulnerability to age-related decline. What this holistic view highlights is the importance of nutrient synergy and metabolic context. Supplementing with isolated vitamins without understanding the broader nutrient matrix or individual metabolic status can yield disappointing or paradoxical results. Conversely, restoring optimal vitamin levels within a well-balanced, micronutrient-rich framework, while modulating energy intake, circadian feeding patterns, and physical activity, can dramatically enhance the body's capacity for autophagic renewal and long-term cellular integrity. In practical terms, this means an anti-aging nutrition strategy must go beyond calories and macros. It should account for genetic polymorphisms that affect vitamin metabolism, such as MTHFR for folate or BCMO1 for beta-carotene, age-related changes in absorption, and environmental factors like medication use or stress that deplete nutrient stores. It also suggests that targeted supplementation, especially of B vitamins, D, A, and antioxidants, should be guided by functional biomarkers, not just population averages. In conclusion, the vitamin autophagy longevity axis is a critical area of aging research that underscores the need for precision, balance, and systems-level thinking. Vitamins are not merely supporting actors in metabolism, they are key regulators of the gene networks that dictate how well we age. When viewed through this lens, autophagy becomes not only a process to be triggered by fasting or exercise, but a system to be nurtured and supported through intelligent micronutrient care. As we refine our understanding of how vitamins regulate these repair pathways, we open the door to a more sustainable and personalized approach to aging well, one built not on restriction alone, but on restoration.